Within living organisms, many chemical reactions are continually taking place, both inside and outside of cells. Most of these reactions could not occur at rates necessary to sustain life without the help of large protein molecules called enzymes. All enzymes in an organism are produced inside an organism's cells. Most enzymes are used to speed up chemical reactions within cells. Some enzymes, however, are passed out of the cells and used in extracellular chemical reactions. A chemical reaction occurs when molecules react with one another to produce different molecules. An enzyme speeds up the rate at which a chemical reaction occurs and is, therefore, a catalyst. A chemical reaction involving an enzyme is said to be an enzyme-catalyzed reaction. The molecules with which an enzyme interacts make up the substrate. In a chemical reaction, the reactants must absorb a certain amount of energy from the environment before the reaction can occur. The specific amount of energy required for the reaction is called the energy of activation. In chemical reactions occurring outside of living organisms, the energy of activation can be supplied by directly applying heat. In a living organism, such as a human, the energy of activation for a chemical reaction cannot be achieved by applying heat. This would raise the temperature within the organism to a level beyond that which sustains life, causing the organism to die. Therefore, the energy of activation must be lowered. This is done by enzymes. Most enzymes have names ending with the suffix "ase". The rest of the name is usually derived from the substrate. For example, the enzyme that breaks down protein is called protease. To understand the way in which an enzyme-catalyzed reaction works, it is necessary to look at the structure of an enzyme. An enzyme is a protein which contains a pocket on its surface called the active site. Each active site is specifically shaped so that only one or two types of substrate can fit into it. By molecular standards, enzymes are enormous. However, it would take over 1 billion of them to cover the head of a pin. There are two different models that describe the mechanism by which the substrate fits into the active site: the lock and key model and the induced fit model. In the lock and key model, the substrate fits into an enzyme active site to form an enzyme-substrate complex. In much the same way, a key fits into its complementary keyhole in a lock. In the induced fit model, the enzyme active site is not exactly complementary to the shape of the substrate. After binding with the substrate, however, the active site changes shape to achieve a better fit around the substrate. After the enzyme and substrate come together to form the enzyme-substrate complex, bonds within the substrate molecule are broken by the enzyme and two smaller molecules are formed. Conversely, an enzyme may also cause two molecules to join. The enzyme brings the two substrate molecules into close proximity of one another so that bonds can form between them. Enzymes are not used up or changed in chemical reactions. Therefore, they can be used over and over again in new reactions. A single enzyme can catalyze thousands of reactions each second. Because of their incredible efficiency, enzymes are only needed in very small concentrations in cells. Many factors have been shown to affect the activity of enzymes. One such factor is the temperature of the environment in which an enzyme functions. As the temperature rises within a cell, the motion of substrate molecules increases. As a result, the substrate molecules come into contact with enzymes much more frequently. Thus, the rate of enzyme activity steadily increases as the temperature rises. Enzyme activity increases steadily with an increase in temperature. In humans, enzymes function best at about 37 degrees Celsius. Notice, however, that when the temperature exceeds a certain point, about 40 degrees Celsius in the case of humans, the rate of enzyme activity begins to decline. This is because enzymes have a tendency to denature at high temperatures. When an enzyme denatures, its active site changes shape. Therefore, it can no longer fit the substrate, making enzyme catalyzed reactions impossible. The rate of an enzyme-catalyzed reaction is dependent on how often enzyme and substrate molecules come into contact with each other. Increasing the concentration of enzyme molecules, therefore, results in an increase in the number of substrate molecules reacting at any moment. Thus, the rate of enzyme activity increases until all substrate molecules are occupied continuously. Once this is achieved, adding more enzymes will not increase the rate of reaction. Increasing substrate concentration when there is less substrate than enzyme also increases the rate of enzyme activity. As you can see, the rate of activity no longer increases once the active sites of all enzyme molecules are occupied. Another factor affecting enzyme activity is pH. The pH at which an enzyme works best can vary greatly from one enzyme to another. Some enzymes, such as the digestive enzyme pepsin, which is found in your stomach, work best in an acidic environment. That is, an environment in which the pH is below 7. Others, however, such as the digestive enzyme trypsin, which is found in your small intestine, work best in a basic environment where the pH is above 7. Shown here is a peat bog. In Denmark recently, a 2,000-year-old perfectly intact body with a noose around its neck was found in a peat bog such as this. Because the soil of the bog is very acidic, the enzymes which normally would have decomposed the body have not been active. The body, therefore, is very well preserved. Many enzymes require one or more non-protein additions, called cofactors, for activity. Cofactors may be derived from vitamins or minerals, such as calcium or iron. If the cofactor is an organic molecule, such as a vitamin, it is called a coenzyme. This explains in part why vitamins and minerals are so important in our diet. The activity of some enzymes can be inhibited by certain inhibitor molecules. A non-competitive inhibitor molecule blocks enzyme activity by binding to an area of the enzyme separate from the active site. Upon doing so, the enzyme changes shape, therefore distorting the shape of the active site and preventing the binding of the substrate molecule. The receptor site that binds a non-competitive inhibitor molecule is called an allosteric site. The non-competitive inhibitor molecule is called an allosteric inhibitor. Enzymes whose activities are controlled by molecules binding to an allosteric site are called allosteric enzymes. The inhibitors that block enzyme activity by occupying the active sites of specific enzymes are called competitive inhibitors, since they compete with the substrate molecules for the active sites. Penicillin is an example of a competitive inhibitor. It blocks the active site of the enzyme that many bacteria use to make their cell walls. Some allosteric enzymes require the binding of a molecule to the allosteric site in order to become active. In this case, the activating molecule is called an allosteric activator. Shown here is an allosteric enzyme made up of 4 protein subunits, each with its own active site. An allosteric site is located at the junction between 2 subunits. The enzyme is constantly alternating between an active and an inactive form. When an allosteric activator molecule binds to an allosteric site, the active form of the enzyme is stabilized, preventing the formation of the inactive enzyme. All active sites of the enzyme are now capable of binding a substrate molecule. When an allosteric inhibitor molecule binds to an allosteric site, the inactive form of the enzyme is stabilized. As a result, none of the subunits is capable of binding a substrate molecule. Substrate molecules are capable of enhancing the activity of enzymes. Recall from the induced fit model that when the substrate enters the area of the active site changes shape for a better fit around the substrate. In an enzyme with 2 or more subunits, the binding of a substrate molecule to one of the active sites induces changes in the remaining active sites so that they become more receptive to substrates. This mechanism is called cooperativity. Most enzyme-catalyzed reactions are actually part of a series of reactions that enables certain compounds to be built up or broken down. Such a series of reactions is called a metabolic pathway. For each reaction in a metabolic pathway, a different enzyme is used. The product of one reaction becomes the substrate for the next reaction, and so on, until an end product is formed. By controlling enzymes, metabolic pathways may be switched on and off. One of the most common methods of metabolic control is feedback inhibition. A pathway is switched off by its end product, which acts as an allosteric inhibitor of an enzyme in the pathway. For example, when the end product binds to the allosteric site of the first enzyme, the shape of the active site is changed so that it can no longer accept the substrate that starts the pathway. This prevents the cell from wasting chemical resources. The importance of enzymes cannot be overstated. They make possible many of the most crucial processes in our bodies, such as cellular respiration, DNA replication, and digestion. Enzymes continue to be the subject of study for many biologists in search of new information that will help us better understand their role in the overall functioning of the cell.